Hello everyone. Today I am discussing the topic about UV visible spectrophotometer. The title says itself that it is depend on UV and visible rays. Let us see what are the requirements first. We need tungsten lamp and hydrogen lamp, monochromator, beam splitter, pair of identical cell and detector. Finally, a recorder that shows wavelength and absorbance graph. Hydrogen lamp. It is used to produce ultraviolet rays. These are range wavelength from 0 to 400 nanometers, and for visible light we use tungsten film lamp. It is range from 200 to 800 nanometers. A monochromator is also used to observe other wavelengths, and what are the wavelength we need is just allowed by it. And we use beam splitter to divide light into two equal parts where the wavelength does not change but the intensity will changes. We use pair of identical cells. Cell is filled with test solution and another with solvent, and these test cells are completely transparent because to absorb the light, and best example is quartz. Detector is used to collect the light and observe the differences and convert into electrical signals and sends to recorder. And finally, recorder will convert the electrical signal to into graph. Okay, let's go for procedure. Hydrogen lamp emits UV light as well as tungsten lamp emits visible rays. They have 0 to 800 nanometers, but 0 to 200 nanometers wavelength, that is UV rays, does not experimentally used. It is used in vacuum only. So we need to convert into 200 to 400 or 200 to 800 between wavelengths. So for that we need to use a monochromator, that is prism. When the beam passes to the prism, it converts from those wavelengths to required wavelength of 200 to 800 nanometers, and it removes or observes all other wavelengths. And one of the beam passes to the solvent that observes all the light and passes the signals to the detector, whereas the another beam travel through the sample and it has some colored solution, so it observes some light and it transfer other light to the detector. Actually this experiment can be done without solvent, then result will come but with some errors. To rectify that errors we need to use solvent. When we use solvent there may some impurities in solvent, but we can adjust the detection and finally we get the best values and the graph. And the detector will collect the light and convert into electrical signals, and it shown as a graph by recorder. And this is the experiment. By this experiment we can know the compound, that is the structure of compound or conjugation or some geometrical isomers, etc. Thanks for watching the video.